This is the fourth part in a five-part series of videos on neutral molecular evolution. For those of you who were wondering when we would actually get back to thinking about neutral evolution, we're ready to do it now. We have all the pieces that we need, so we're going to put it all together and look at why Kimura in particular felt that most changes in amino acid sequences were probably neutral. All right, let's remember now what we've done. We've defined a rate of evolution, which is the rate of change in a protein between a pair of species. We've talked about genetic variation, and we talked about two different measures of that. We talked about the proportion heterozygosity and the percentage of polymorphic loci. So we've got those two major types of measures of genetic variation of one form or another. We defined genetic load, and we talked about what the cost of selection was and the fact that selection can't be too strong or else it's going to impose such a strong load that a population will be driven to extinction. All right, so now let's look at what Kimura did with this. He looked at it and he came to two conclusions. One, he decided that the rate of molecular evolution, or the rates of molecular evolution that were being measured, were way too fast to be explained by selection. He also looked at the amount of genetic variation that was being found in populations, and he decided that based upon the evolutionary theory of the day, that there was way too much genetic variation in populations also to be explained by selection. And when he saw this, he thought, well, maybe some of the variation that we're seeing, or some of the rates, are actually due to the variation being neutral. So let's take a look at the details on this. All right, so his first claim was that the rate of molecular evolution was simply too rapid to be accounted for by natural selection. How did he come to this conclusion? Well, the earliest estimates of the rates of substitution for proteins were way too high considering Haldane's estimates. Remember, Haldane said that he thought that you could only substitute one amino acid about every 300 generations, and if you did it faster than that, you were going to end up killing off the population due to too big a genetic load. All right, so let's look at what did Kimura know. He knew, for example, that the average human genome size was about 3 billion nucleotides. Okay? Now, what he didn't know back in 1968 was that there was a lot of non-coding DNA. So he assumed that most of the DNA probably coded for something, and therefore, since there are 3 codons per amino acid, that there would be 1 billion amino acid sites in the human genome, for example. Now, the average rate of substitution that had been measured was about 3.57 changes, amino acid changes, per site per year. Okay? Now, why is this problematic? Because if there are 1 billion sites, and we multiply that 1 billion amino acid sites times the average rate of change per site per year, it means that on average, over 1/3 of a site changes per year. Another way of putting this would be to say that it takes less than 3 years for one site to be substituted somewhere in the genome for another amino acid. All right? Now that's way too fast. Remember, Haldane thought you could only have about one substitution every 300 generations or years. And instead what we're seeing are one change every 3 years. So that makes the rate of substitution about 100 times faster than Haldane had estimated. This would impose an enormous genetic load on a population. And so Kimura looked at this and thought, this is impossible. If, these are the data that we have for the rates of substitution, and they're just way too fast. So I can't believe that all of this substitution is taking place simply because of selection acting on these populations. Okay, so the rate of substitution is too fast for selection to be operating. Now, on the other hand, if the variation was not subject to selection, it was just all there due to drift, then there's no cost to that. There's no death taking place. You're just getting random changes in frequencies of alleles like you learned about in the first half of the course. So since nobody dies to change the gene frequencies under drift, there's no cost. And so there's no problem with having this rate of substitution occurring in those populations. All right, what about his second claim that the amount of genetic variation was too high? What did he see? Well, people have done lots of gel electrophoresis of proteins in many, many different populations. And so it seemed like there was too much variation within populations for selection to be the cause. Let's look at one example from Drosophila. So in Drosophila, population variation may run as high as 30% polymorphic loci. So that means that 30% of the loci looked at had at least some variation. And on average, they would find something around 10% heterozygosity at an average locus. Okay? Now, if we know that that's true, then what happens? Well, again, this is back in 1968. Nobody really knew exactly how many independent genes there were in fruit flies. But at the time, it was estimated to be about 10,000, and that wasn't too bad of an estimate, really. And so if 30% of those loci are polymorphic, right, that's what this says up here, that would mean that of these 10,000 loci in the fly, 3,000 of them would be considered polymorphic, okay? Then the question becomes, all right, so what's the average heterozygosity of just the polymorphic loci? Now think about this for a minute, because this is a little bit complicated. You say, well, wait a minute, don't we already know what the average heterozygosity is? It's 10%. Well, yes, we do know what the average heterozygosity is, but the way that we calculate the average heterozygosity is to take all of the loci, whether they're polymorphic or not, and take an average of those. So many of those loci, in fact, according to this estimate, about 7,000 of them, did not have any heterozygosity at all. So what we really want to know right now is not what is the average heterozygosity of all of the loci, but just what is the average heterozygosity of the loci that have some amount of heterozygous individuals at all, okay? So that's what we're doing here in this next little part. So what we want to know is what's the heterozygosity of just the polymorphic loci? And that turns out to be about 33%. And the way that you get this is you just take a ratio of the average heterozygosity and divide that by the percentage of polymorphic loci, and that gives us the average heterozygosity of the polymorphic loci, or 33%. All right, why does all this matter? Let's look at the next slide. Here's the big deal. In Kamiro's time, it was thought that the only way, or at least the primary way, of maintaining any kind of polymorphism in a population was by heterozygous advantage, or overdominance. Okay, now you've already learned about heterozygous advantage in an earlier part of the course, so I'm assuming you remember all this. Now, it's not a big deal for selection to be maintaining variation for just one locus by heterozygous advantage. But it doesn't work very well if you've got 3,000 loci that are all being maintained in some degree of heterozygosity by natural selection. So let's take a look at this. So let's assume that selection against the homozygous types is about 10%. And we've already said that the average heterozygosity of one of these loci is 33. What that means then is that the average fitness in the population under these circumstances is going to be about 96.7%, or 0.967. Okay? If you want to think about it in this way, the population is never going to reach an optimal fitness when you've got heterozygous advantage, because you can't fix one of the alleles when you've got heterozygous advantage, right? The whole point of heterozygous advantage is that both of the alleles are being maintained in the population. So there is a slight genetic load that's being imposed on that population that will never go away due to the heterozygous advantage. So it would just be 1 minus W, or 1 minus 0.967. Now that's not a very big genetic load, right? We're talking about a genetic load of about 3%. But here's where the real problem comes from. When you've got 3,000 heterozygous loci, and if each one is independently affecting the fitness of the population, to get the overall effect on the average fitness of the population, we have to take the effect on a single locus and raise that to the number of loci. So we take 0.967 and raise it to the 3,000th power, and that is approximately 10 to the minus 43. Okay, this is what we call multiplicative fitness. Now let's think about what that really means. 10 to the minus 43 is a very, very tiny number. Okay, almost zero, but not quite. And so basically, the genetic load has become enormous in this population because 1 minus approximately 0 is still going to be approximately 1, giving this population a genetic load of about 1. And so Kimura looked at that and thought, that's just simply not possible. In fact, another researcher named Dick Lewontin came up with a very pithy phrase, which is, one can hardly imagine a Drosophila female, no matter how many loci she was heterozygous for, laying 10 to the 43 eggs. And the reason that he said it this way is because if the population has a genetic load of 1 minus 10 to the negative 43 power, it means that if a female was able to survive to adulthood in this condition, she would have to lay at least 10 to the 43 eggs to ensure that at least one female would survive to adulthood in the next generation, so she could then go on to lay 10 to the 43 eggs. No organism is ever going to lay 10 to the 43 eggs. In fact, 10 to the 43 is so big it's larger than the number of fundamental particles in the entire universe. So you can see why when Kimura did these calculations, he ended up saying, okay, whoa, wait a minute. We're not maintaining all of these heterozygous loci by natural selection. If that were true, then nothing would be alive. And so he says, well, maybe that means that a lot of these loci are simply ones in which there is no selection operating, that it's just subject to drift, and therefore we can have so many of these because of that.